Hello, Chem 213 students, and welcome back for this week's lectures, this time on Carbohydrates, Chapter 24. Today is going to be the first lecture where we're finally going to take advantage of all the organic reactions that we've learned and apply them to biology. And we're going to start to look at a variety of biochemicals here, and namely the most important macronutrients, carbohydrates, proteins, and lipids. And we're going to see how the reactions and the chemistry that we've learned that are not in biological systems actually applies in much the same way to biological systems. It's the same reactions occurring. The only real difference is their kinetics can be improved, or the reactions can be made faster by introduction of enzyme catalysts, which are proteins that make reactions faster. But otherwise, the reactions are the exact same reactions. So we're going to see now how the organic chemistry you learned applies to biology, starting with carbohydrates, which includes simple sugars like table sugar here, which is referred to as a disaccharide, or like bread, which contains starches with many carbohydrate molecules chained together. So for this chapter, we're going to be drawing upon many of the things we learned previously, and that's going to be true throughout this biochemistry, this last biochemistry section of this class. In this particular chapter, we're going to be looking very much at cyclohexane type conformations. So chair conformations will be important. Haworth projections, Fisher projections as well. So these different ways of drawing molecules are going to become very important for analyzing large carbohydrate molecules. And so you want to have a firm foundation in Chapter 4 and Chapter 5. In addition, carbohydrates are going to form into ring structures by a hemiacetal-acetal reaction that carbonyls, ketones and aldehydes undergo. And so we're going, if you've forgotten that, that's just from, earlier this semester. Be sure to go back and check section 19.5. And I would take a second look at that mechanism of the acetal formation before you begin this chapter, and make sure that you've got that down quite well, because we're going to kind of gloss over it because we've already learned it previously. But it is going to apply here in this chapter. In addition, carbohydrates can be reduced to sugar alcohols, and these are the kinds of sugars that you commonly find in, for example, sugar-free chocolates. They very often have maltitol or something like that, erythritol, which are sugar alcohols that are made by reducing sugars, and then that makes it so your body cannot digest them, and so it passes through you without being digested, and you don't get any calories from it. Or it's digested a little bit, but not as much as sugar. So where do these sugars come from? Carbohydrates, which are also called sugars. So you've been noticing that I use the term kind of interchangeably. Sugars are basically simple carbohydrates, small carbohydrates. These are polyhydroxyaldehydes or ketones. So what is meant by polyhydroxy? It means that the molecule contains many hydroxyl groups. So when you look at glucose here, for example, which is blood sugar, you can see that every carbon, except for this one with the aldehyde, every other carbon that is not this aldehyde has hydroxyl groups on it, either on a wedge or a dash. So we say polyhydroxy, many hydroxyl groups, aldehyde or ketone. And so also the word carbohydrate comes from the idea that at one time they were thought to be hydrates of carbon, meaning that if you look at every carbon here that is not the aldehyde, it will have one hydrogen attached and one hydroxyl group attached. So together that's, two hydrogens and one oxygen, water. It says a water attached to each carbon, basically. So this is where the word carbohydrate comes from. Now we know the actual structures of these. We know they aren't hydrates of carbon atoms. They're actually polyhydroxyaldehydes and ketones, but that's where the traditional historical name for carbohydrates comes from. And the way carbohydrates are formed is in a complicated photosynthesis process that we are still learning more about and still do research about. But in photosynthesis here, CO2 is combined with water to produce a carbohydrate and oxygen. And so this is the photosynthesis process that you might learn more about in biology, but this is the way that carbohydrates are made biologically. So monosaccharides are what we would call simple sugars. So a monosaccharide is one that contains a single molecule of a polyhydroxyaldehyde or ketone. So an example of a monosaccharide here is the glucose we just saw. And you can see that it's written in a Fisher projection. So remember, Fisher projections are written in such a way that the sides here, these are all wedges, right? These are all wedges, and they're mild wedges. And then these are all mild dashes, meaning that if I had a tetrahedral shape, I'd be pointing two at you, and then there would be two sticking back, right? And so that's the formation. And really, in real life, it would be hard to build this with a model exactly as the Fisher projection, because all of these on the vertical axis would be going back, right? So it kind of wraps around in on itself. But it's a really great way to analyze the stereochemistry of a molecule that has many, many chiral centers. And we can see that all of these carbons are right here, right here, right here, right here. There are 4 chiral centers on glucose. And so using a Fisher projection is a great way to keep track of where those are, so because we can just look at them as being here on the right side of the Fisher projection or on the left side. Right, and so it's much easier to analyze than a straight chain structure like this. So notice this is the same straight chain structure here, that I'm referring to. We've got 3 on a wedge and 3 on a dash, 1 on a wedge, and you can see that this doesn't it's hard to translate between these. I don't tend to make you guys do that. You can see with models, you can kind of figure it out, but it's a little tricky. But anyway, for fructose here, this one also has 3 chiral centers right here, and right here. And so this Fisher projection is a great way to represent their structure, and it's easier to analyze than a structure like our standard structure here, because we're really paying attention to the chirality, the stereochemistry of each carbon here. So carbohydrates are classified broadly into 2 main categories, and those are aldoses and ketoses. So aldoses are polyhydroxyaldehydes. So they are structures in which each carbon has, like this, we have this OH and H group here, polyhydroxy carbons here, and then on the carbon at the end we have an aldehyde. This is referred to as an aldose. Ketose here, similarly, instead of having an aldehyde as a ketone, and that ketone always appears on the second carbon here. So there's one carbon with a hydroxyl group on the end, then we have the ketone group here, and then we have the remaining polyhydroxy part here as we go down. So these are the 2 main categories. They either have a aldehyde or a ketone that's on the second carbon. It's not in the middle of the molecule. It's always on the second carbon. And then we use a prefix that indicates how many carbons are in there. So in the case of this molecule here, it has 1, 2, 3, 4, 5 carbons, and it's an aldehyde. So we'd begin by describing it as an aldose. And if we wanted to describe specifically how many carbons, we'd call it an aldopentose. And so by putting a pent here between aldo and os, or an os here, this tells us that it's an aldose with 5 carbons. Here in this case we have a ketose. We can see the ketone group here. It has 1, 2, 3, 4, 5, 6 carbons. So it's a ketose because it has 6 carbons. And if we want to be specific that it's 6 carbons, we call it a ketohexose. So ketohex. So we put hex right there between the ket and the os. We call it ketohexose with 6 carbons. And then, besides classifying them these carbohydrates based on whether or not they have an aldehyde group or a ketone group, and based on how many carbons they have, we also can classify them based on their stereochemistry, And the stereochemistry has an interesting history where it is based off of the simplest carbohydrate, glyceraldehyde. And this is because historically these carbohydrates were broken down to glyceraldehyde by chemical reactions when they were studied. And it was noticed that natural sugars, when you broke them down, only produced D-glyceraldehyde, or they only produced the R version. So this happens to be R, and this happens to be S. And so when the natural sugars were broken down, they only had the R version. And that R version of glyceraldehyde rotates plane polarized light in the clockwise direction. That's what the plus means, right? Whereas the S-glyceraldehyde rotates plane polarized light in the counterclockwise direction and is thus indicated as negative. We also use this terminology, D or dextratory, meaning that plane polarized light is rotated clockwise, versus L, levratory, or left, where basically the plane polarized light is rotated counterclockwise. Now, the interesting thing is that this D or L designation is applied to carbohydrates regardless of their rotation of plane polarized light, okay? So that makes it a little bit confusing. You might think that every carbohydrate labeled as D would be positive in terms of its rotation of plane polarized light, but that is not the case. It's only referring to basically the location of the hydroxyl group on the last carbon before the end, furthest from the aldehyde or ketone group. And so they're all based upon glyceraldehyde. All D sugars thus will have the OH group positioned on the right in the Fischer projection on the bottommost carbon here. And the other ones, that's not going to affect whether it's called D or L. Just the last carbon here. And this is because, again, the D or L designation for carbohydrates is different than for other molecules. It's only applying to the stereochemical configuration of the hydroxyl group on the last carbon. And these are some common organic sugars here, biological sugars, and you can see they are all of the D configuration, where the hydroxyl group is on the right side on the very last carbon. And L configuration carbohydrates are not common in nature. They're very uncommon. So again, I must emphasize that D and L in terms of carbohydrates mean something different. They do not necessarily mean that the plane polarized light is rotated clockwise, or that there's a positive rotation of the plane polarized light. For example, D-erythros and D-threose, these both have negative counterclockwise rotation of plane polarized light, even though they are designated as D. They're designated as D only because the hydroxyl group on the last carbon here before the end, the last chiral carbon, is located on the right side in the Fischer projection. And this is, a reference back to the R version of glyceraldehyde versus the S version. Okay. So again, the take-home message: D sugar means the configuration of the furthest chiral center is R, while the L sugar has the S configuration at the furthest chiral center. So if we have, going back here to glyceraldehyde, there are only two stereoisomers of glyceraldehyde, because there's only one chiral carbon right here. And so there's the R and the S, and that's it. But if you have two chiral centers, now you have 4 total potential stereoisomers. Okay, and here, so with erythreos, we can have D-erythros rather, and D-threose. Here it would be the one where the hydroxyl group is on the left side. So notice these are both D. They are both D because the bottom carbon here has the hydroxyl group positioned on the right side. However, D-threose is the diastereomer of D-erythrose. Okay, it's not the enantiomer. The enantiomer would be the mirror image. So, for example, if we wanted to have L-erythrose, or erythrose, we would simply reverse the stereochemical position of each of these. And so the L version here would be OH on this side and also OH on this side. And this would be L-erythrose. So these are enantiomers of one another. And these here are diastereomers of one another. Right? And threose has its own enantiomer. If you want to draw the enantiomer of threose, you simply, again, reverse the stereochemical positions of each of these OH groups. So put this OH group on the right and put this OH group on the left, and then you would have L-threose. Now, if you have 3 chiral centers, you have 8 possible stereoisomers. So the way you get, if you remember from chapter 5, the way you get the number of stereoisomers is you take the number of chiral centers and you do 2, so the total stereoisomers here are going to, it's 2 to the second power being the number of chiral centers. Now you have to be careful because this is to be exact, this is the max number of stereoisomers. There could be fewer if any of these are meso, right? So that's something to be careful of. In the case of carbohydrates, though, they will not be meso because there will never be a plane of symmetry, because there will always be an aldehyde or ketone group on one side and you will not have one on the other. So we don't have to worry about that. In this case, we're going to have 4 stereoisomers. We'll have erythrose, threose, and their 2 enantiomers. Now here are the 4 different diastereomers we can have when we have 3 chiral centers. So all of these have 3 chiral centers, and they're all D. They're all D versions of these. Okay, they have a D designation because the OH group on the last carbon here is positioned on the right side. Okay. However, we have other diastereomers because we can move the location of the other OH group. So if we take the OH, so D-ribose has all of the OH groups positioned on the right. And the fact that the last one on this carbon here is to the right indicates that it's a D sugar. Now, if we take this hydroxyl group and we switch its position here, then we get D-arabinose. Okay. If instead we start with D-ribose and we take this OH group and we move its position over here to the left, we get D-xylose. And if we take both these hydroxyl groups and we move them to the left, we get D-lexose. And then that would be all the D sugars, and it's only D sugars that are naturally occurring. So we're not as concerned with the L sugars. We can get them. We can get the L sugars just by taking all the rest here. And if we want L-ribose again, we take all these hydroxyl groups and we switch them over to the left side. We get D-ribose. We want our L-ribose rather. If we want L-arabinose, we take all the hydroxyl groups and switch their side left and right, and we get L-arabinose. And so we have here 8 possible stereoisomers, each of these D sugars and their enantiomers. And that is because there are 3 chiral centers. So we have 2 to the 3rd power. So these are all aldopentoses. They have aldehyde groups, and they have 1, 2, 3, 4, 5. So these are all 4 D-aldopentoses, and then the L-aldopentoses would be their enantiomer. But again, D sugars are the ones that occur naturally, so we're the ones that we're most concerned with. Now, I'm not going to force you to memorize these names as well. I don't think that's particularly important, but I won't make you do the kinds of analysis that we are doing right here. Aldohexoses will have 4 chiral centers. Thus, the number of chiral centers will be 2 to the 4th power, which is 16. So there will be 8D isomers and there will be 8L isomers, okay, which are the enantiomers of all the D isomers. So again, we're going to focus on the D ones, the D stereoisomers, because those are the ones that are naturally occurring. And so the 8 naturally occurring D sugars are listed below. Again, a lot of names that you would, if you take a biochemistry class later on, they might make you memorize. I don't think it's important for you to memorize all of these names. Perhaps the only one that you really might want to kind of have memorized, and you might end up memorizing a little bit, is glucose. That's blood sugar. That's most important for mammals. It's very, the one we're going to see a lot and pay a lot of attention to because it's our blood sugar. So again, here we have 8 isomers, and we can figure out all the isomers. These are all going to be diastereomers of one another. We can figure out the 8 diastereomers here simply by methodically going through every possible position of hydroxyl groups except on the last carbon, because it always has to be on the right for it to be D. So altrose, we just take the top hydroxyl group and move it to the left. Glucose is just taking the second one, moving it to the left, switching its spot. Mannose is taking both the top 2 and switching them to the left. Gulose is taking the third hydroxyl group and switching it to the left here. Aldose here is taking the top one and the bottom one and switching it to the left. Okay. Galactose is taking the middle 2 here and switching them to the left. And then talose is all hydroxyl groups except for the last one on the left. So galactose, that's another one that will be important too, because this is going to be one of the sugars that is in lactose, which is a disaccharide. We'll talk about disaccharides a little later. They're molecules that have more than one carbohydrate molecule stuck together, bonded together in what's called a glycosidic linkage. So here's a overall review of all of the aldoses, and I know this is probably going to be small on the screen here, but it's just covering the same things we did. And these are all the natural aldoses, all the D sugars. So if we have this is going to be these are Aldotetroses, and there's only one because there's only one stereochemical center here. It's D-glyceraldehyde, and its enantiomer would be L-glyceraldehyde. Here we have the Aldotetroses. Then we have the Aldopentoses. Then we have the Aldohexoses. And again, in terms of the number of, so this has 1 chiral center, so I'll just say chiral centers. So it has 1, this has 2, this has 3, this has 4. So your number of stereoisomers is 2 to the 1, so that's 2. So we have this and it's an enantiomer. Here's 2 to the 2, so we have 4. We have these 2 and they're enantiomers. We have then 2 to the 3rd, that's 8. So we have these 4 sugars and their enantiomers. And then we have 2 to the 4th, which is 16. So we have these 8 sugars and their enantiomers here. And so that is the full collection here of natural aldose sugars. There are also many naturally occurring D-ketoses. So here, as we go up in this diagram, it's a similar type of thing. So let's analyze it the same way we did the aldoses. If we have here a 3-carbon ketone, well, this is not going to be a carbohydrate at all because there will be no chiral centers. There'll be no hydroxyl groups except those in the end. It's just hydroxyl group here, and a ketone in the middle. So this is just called dihydroxyacetone. It's like acetone, but 2 of the hydrogens were replaced with hydroxyl groups. So it's called dihydroxyacetone as a common name. So the first naturally occurring D-ketose that we'll have will contain 4 carbons. So this is going to be a ketotetrose. 4 carbons, 1, 2, 3, and 4. And then these will contain 5 carbons, 1, 2, 3, 4, 5. So these are going to be ketopentoses. And see, these ones will contain 6 carbons, so these are ketohexoses. Now, there's only going to be one enantiomer of the ketotetrose, and it's because there's only one carbon, this third carbon right here, that is chiral carbon. This is not a chiral carbon. It has 2 hydrogens attached. This is not a chiral carbon. This has 2 hydrogens attached. This carbon is part of the carbonyl bond. It does not have 4 groups attached because it has a double bond. So the only carbon with 4 groups attached is this third one. And if we want the D enantiomer, this will be the last hydroxyl group. It's the only one. So having it to the right makes it the D enantiomer. For the ketopentoses, those will contain 2. So here there are 2 to the 1 stereoisomers rather. So there's going to be 2. There's going to be D erythrulose, and it's an enantiomer. And then for stereoisomers here, there'll be 2 to the 2, which is going to be 4. You'll have these 2 diastereomers, and each of them have their own enantiomer, which is the L form. So D-ribulose has the 2 hydroxyl groups on the right side. D-xylulose will have this first hydroxyl group here on the left. And of course, both of them are D because their last hydroxyl group here is on the right. And then ketohexoses will have 3 chiral centers, 1, 2, 3. And their number of stereoisomers will be 2 to the 3rd, which is 8. And 4 of them are L enantiomers, 4 of them are D enantiomers, and so these are the 4 D diastereomers of one another. The one with all the hydroxyl groups on the right here is called D-cyclose. If you take the top hydroxyl group and you move it to the left, this is D-fructose. D-fructose is one of our most important ketoses because it is a part of sucrose. So it's in sucrose. Sucrose is a disaccharide that is table sugar. And so table sugar is made of glucose and fructose, 2 carbohydrate monosaccharides bonded together. And so fructose is another one that you're going to encounter a lot. So here the OH group is positioned on the left, both of the bottom ones here on the right. Here the second hydroxyl group is positioned on the left, while the first one remains on the right. This is called D-sorbose. And then if both hydroxyl groups in the top 2 carbons here are on the left, we'll call that D-tagatose. And again, the last hydroxyl group is always on the right to make it D. All of these have an enantiomer where the bottom hydroxyl group here is on the left, and those are the L enantiomers of each of those. Now, although we've shown as Fisher projections these ketoses and hexoses in the form of a straight chain molecule, in normal conditions, normal aqueous conditions like in your bloodstream, these will primarily exist in a ringed form that is due to a acetal reaction, hemiacetal reaction, that occurs when the hydroxyl group reacts with the carbonyl group in its ring. So you may remember from chapter 19.5, and again, I encourage you to look again at that mechanism there. But here you're going to get the formation of the hemiacetal, which essentially is going to add on a alkoxy group here. And the alkoxy group can be whatever. In the case of the sugar, it's its own structure. So you may recall here the carbonyl gets protonated, and then that is followed by a nucleophilic attack by the alcohol, and then ultimately we produce the hemiacetal, right? We get the OH. So it starts like this. It's 2 steps. First, the hydroxyl group gets it gets protonated, and so it has a positive formal charge. Then the nucleophile can attack it, makes it a better electrophile here, and then we get the formation of an alcohol group here, and we get then the deprotonation here of this guy, and then we'll get the OR group. So go back and review that mechanism. That's a very important one just to remember in general. But in the case of a molecule that contains its own hydroxyl group, it will be its own hydroxyl group that ends up doing the nucleophilic attack on the carbonyl itself. And so, or in the case right here, sometimes it's useful to kind of write this around in a ring. So if we call this carbon 1, 2, 3, 4, 5, this will be carbon 1, 2, 3, 4, and 5. And so we've drawn it around in a ring to show now the proximity of the attacking hydroxyl group here. Okay. And that puts the oxygen here at the upper right hand of the ring, which is the traditional place where people regularly put the oxygen in a carbohydrate to kind of, so that everyone's kind of drawing it the same for easy comparison. And so that's the meaning behind kind of wrapping around in this direction to put this oxygen in the top right, because when this ring closes, which it does here when a bond is formed, this ring closes, and then this ends up making a hydroxyl group when it's protonated. Then we get the cyclic hemiacetal. And this is just like the reaction we saw in 19.5. So if we want to do this for carbohydrates, we do the same thing. We number the carbon here. So carbon number 1 is going to be the aldehyde, and we're going to want to wrap this around so that the OH group gets positioned in the top right. So we have carbon number 1 here. This is carbon number 1. So this is going to be the aldehyde group, carbon number 1 here. And then we have carbon number 2. This is the next one here. And what you're going to notice is that in carbon number 2, what we've done is we've drawn the hydroxyl group in the down position. Okay, and this is going to be the customary way of writing these particular isomers. If it's on the right side, if the OH is on the right side, we're going to draw it in the down position on the ring form, if we're doing it in this style, right? And if it's on the left side of the Fisher projection, we're going to write it in the up position, okay? So here we have a glucose, and it's going into a 6-membered ring. So on the second carbon, we've written the hydroxyl group in the down position. Notice that on the third carbon, which is right here, the hydroxyl group is on the left, which means we're going to write it in the up position on this ringed form. Okay. On the fourth carbon, our hydroxyl group is on the right side, so we're going to draw it into the down position. Okay. And then the alcohol group that's going to be reacting in this glucose is going to be the one on the fifth carbon. So on the fifth carbon, that's going to be the OH that reacts. So that's what we've drawn in red here. It's the hydroxyl group on the last chiral carbon that's going to close the ring in this aldohexose. Finally, the sixth carbon is going to be hanging out here in the up position, which is where we traditionally draw it. So this one's going to be in the up position, this last carbon here. And now we're going to have that and there's a little bit more to this. I'm not drawing the full mechanism, but essentially what's going on is we're going to have this nucleophilic attack, right? And then we have protonation. Really, the protonation happens first, but the result is that this OH group closes the ring. So our alcohol here, now having been drawn right here, ends up at the top right position, which is the traditional place to put it. And then this hydroxyl group here gets protonated, it loses a double bond, it gets protonated and makes a hydroxyl group in the acidic conditions. And then we get the closed form. And again, in the closed form here, we put this guy right here, the sixth carbon, we put it in the up position. And the other hydroxyl groups, we put them in the position such that if it's on the right, we draw it in the down position on the closed ring. If it's on the left, we draw it in the up position. And the equilibrium is going to greatly favor the cyclic form. So when this blood sugar, this glucose, is floating around in your body, it is generally forming around, the massive majority of the molecules are in this closed ring form. And this resembles this is called a pyranose because it's got a ring here that has 5 carbons and 1 oxygen. So it's similar it's similarly named to the molecule that has that oxygen in there with a six-membered ring. Now, there are two possible ways that this ring can close. So you can imagine that the ring can wrap around in this direction that we showed it, or it could wrap around in the other direction. And depending on which direction it attacks, so depending on whether the nucleophilic attack comes from one side or the other, this hydroxyl group here will either be in the up position or the down position, the hydroxyl group that's formed from the ketone. And so these two, the up position or the down position, these rapidly interconvert. As the ring opens and closes, it can form either one of these. And so these are referred to as anomers of one another. And in the alpha anomer, the OH group is going to be trans to the CH2OH group. And so by keeping this up, we make a constant reference to this guy here. And so we keep this the same in the up position. We change this one as up or down. That's a kind of standardization to make sure that we don't get confused. And then when the OH group is positioned on the down bottom, we call it the alpha anomer. And when it's positioned in the up direction, same as this group here on the 6th carbon, the hydroxyl group in the 6th carbon, we call that the beta anomer. Now you might wonder, why do we care about this? It's actually extremely important. What we're going to see is when these alpha anomer and beta anomer, when they react and produce sugars that disaccharides, polysaccharides, when they produce carbohydrates that are formed from many of these individual ringed units that will be linked together, that the way in which they link at the alpha configuration or the beta configuration is going to make a huge difference in the way that they are processed biologically. For example, human beings, we can very readily eat starches and disaccharides and other complex carbohydrates that are composed of alpha bonds between multiple units here. Whereas, and that's called starches, but when there are beta linkages, as we're going to call, those cannot be easily digested by humans for many cases and by many mammals. So, as I said, these two forms can rapidly interconvert. Okay, the closed chain form is going to be 99.99% of them, but when it closes, it can close again in one direction or in the other. Okay, this carbon is not chiral. This hydroxyl group can rotate. So depending on where it is when the ring closes, depending on the direction of the attack, you're going to get either version here. So here in this case, we have the alpha D-glucopyranose. So this is what we call it. This is glucose. We call it glucopyranose when it's in the ringed form. We say D because this hydroxyl group is always on the right side. It's D for natural sugars, and alpha because this OH group is on the opposite position as this carbon group, the 6th carbon here. Whereas we call it beta D-glucopyranose when in the ring form the hydroxyl group is on the same side of the ring as the 6th carbon. And these can interconvert because although a very small percentage of any of these straight chain molecules exist at any one time, these are very rapidly, occasionally breaking open and going back to the open form, which can then convert into the other ringed form by closing again. So this is what's referred to as a dynamic equilibrium. So these can convert in between each other and the alpha and beta anomers, and this conversion is referred to as mutarotation. If you have acid or base present, this mutarotation will occur more quickly because the opening and the closing of the ring will be catalyzed and will occur much more quickly. With some sugars, the alpha form dominates, but the beta anomers for others, and it's hard to tell which will be which just by looking at the structure. You'd have to do a kind of more complicated quantum mechanical analysis to say something about that. You can't just tell just by looking at it. When it comes to drawing the Pyranose rings, as I said, when you draw from the Fisher projection for the going back here for the OH that appears on the right, right of the Fisher projection, it goes to the down position. OH is appearing on the left, go in the up position. The oxygen group should be up at the top right. Now, you may also want to consider the actual more realistic three-dimensional form. So we know that six-membered rings like this, we know from chapter 4, that they don't commonly exist as flat rings. They don't exist as flat rings. They pucker, they bend to get lower ring strain, right? And for six-membered rings, they go into chair conformations, being the most stable conformation, like a cyclohexane. So we can draw this like a cyclohexane. When we do, it's traditional, again, to put the oxygen in the top right position here. And one thing that you're going to notice, now glucose, I said, was a very important sugar, right? And it's kind of the nature's sugar of choice for carrying the energy around our bloodstream. So a great question is why? Why glucose and why not some other sugar? There's actually a beautifully simple answer to this if you understand the chair conformations from chapter 4 very well. And that is that if we have this sugar here, so we have glucose, we have the beta form in this case, but if we look at this beta form, we see that we're going to go around the ring here, okay? So we're going to say, okay, we're going to draw this chair form. So here's a chair form, and our axial positions, I'm going to draw in all of our axial positions in red. So this is going to be the foot. Again, we're traditionally putting the oxygen right here. So we're going to make this one, this carbon here is going to be the foot, and this one's going to be the head. We could do the opposite too. We could chair flip it. But here, when we put this on the foot, that's where we get the interesting result. So the axial position on the foot is down, and the axial position on the head is up. Then the axial position on the ring goes up, down, all the way around the ring. So the axial position here is, since this is down, this one's up, this one's down, this one's up, and this one is down. So what you'll notice here is that the OH groups here, this OH group is in the down position, and down here, up is axial, so down is equatorial, right? And we've drawn it here, the down equatorial position. On the next carbon, this carbon right here, OH is in the up position. Up here, down is axial, so up is equatorial. So we've drawn the OH in the equatorial position here. Remember, that is parallel to this line right here. This one is parallel to these right here. On the next carbon here, the OH is in the down position. Well, on this carbon, the up position is axial, so the down position is equatorial. The OH group is once again equatorial. And then if we look at this last carbon here, this carbon is in the up position. The down position is the axial position. So the up position is equatorial again. So all of our hydroxyl groups and our CH2OH group here are all in the equatorial position, making the ring form of glucose spectacularly stable. And that is the reason for evolution's choice of glucose, which I think personally is extremely beautiful. And there's going to be other occasions like this where all these reactions you're learning about, all this weird stuff, then you look at the way biology used it and you see, whoa, this actually, all these ideas of, oh yeah, these groups are more stable if they're in the equatorial position because they're sticking out and there's not steric hindrance. All that stuff that kind of seemed like, oh, this is only, in the chemistry lab, I care about this. No, biology cares about this. Biology is using these properties to make an extremely stable sugar. That means that it's, for example, photosynthesis can make more easily and make more spontaneously because it's making something extremely stable. And so the products have a low Gibbs free energy, and so you get this spontaneous reaction. So it's so cool, I think, that we can use our understanding of organic chemistry to understand the way evolution occurred and the way where carbohydrates came from in an evolutionary sense. The most stable sugar and the most abundant in nature because of that. Now, we can also have Furanose rings when we have a hemiacetal with a with a ketose rather, with a ketose. So ketones can also be involved in hemiacetal reactions, and so we can have a hemiacetal reaction with the ketone. Here again, the hydroxyl group that's going to behave as a nucleophile in this reaction is going to be the last one on the last chiral carbon. The carbonyl that was going to be the electrophile and be attacked by the nucleophile will be the aldehyde. Now we're going to do the same thing we did before. We're going to put the oxygen here on the top, not the top left in this case because it's not 6-membered, it's 5-ring membered. So we're going to put it in the top here. So our first carbon is this one right here. Our second carbon is now the ketone, which is going to be involved in the reaction. The third carbon, it has the hydroxyl group on the left, so we're going to put that in the up position here. Okay. The fourth hydroxyl group has the OH group on the right, so we're going to put that in the down position. And the fifth hydroxyl group is going to be the one involved in the nucleophilic attack on the carbonyl. Okay, and so that's one form of the reaction. I'll show you there's actually two. You can form a Furanose and a Pyranose. But when this occurs, okay, again, I'm simplifying the mechanism here. It's not the full mechanism, but part of the mechanism is we get a nucleophilic attack, right? It's actually protonated first, but so what this means is the ring closes, so now this oxygen group is attached to this carbon, ends up being deprotonated in the end. Here this oxygen gets protonated and becomes the hydroxyl group, and then we get a 5-membered ring with the oxygen. And again, comparing to the Fischer projection, the OH on the left here ends up in the up position. The OH on the two OHs on the right here, or the OH on the right here on the fourth carbon ends up in the down position. The OH on the fifth carbon was the one involved in closing the ring and making this ether right here. Well, now in the ketoses here, not only can the fifth carbon react here, also the sixth carbon hydroxyl group can react. And so we're going to have the same type of thing. We're going to wrap this around. Now it's this last hydroxyl group on the sixth carbon that's going to be involved in the nucleophilic attack. That's the one that's going to close the ring. In terms of the hydroxyl groups, again, third carbon left up, fourth carbon right down, fifth carbon right down, and sixth carbon, that's the one involved in the nucleophilic attack. So that's going to close the ring with a bond between this oxygen and this hydrogen. Of course, it ultimately is deprotonated, and the oxygen here is no longer in a double bond, and it is protonated. The hydrogen ends up over there. And again, one more time, left is up, right is down in the ring. So left, up, OH group, on carbon 4 and 5 to the right, down. And then the last OH group here, this oxygen is involved in this ether right here. And so that's the way you take a Fischer projection and turn it into the ring form, which is really the more true form, the form that these monosaccharides usually exist in. So the Furanose form of D-fructose takes part in most biochemical reactions. So although fructose can make either of these, when we see fructose, this is fructose. When we see fructose present in sucrose, for example, table sugar, it is taking this more common Furanose ring form. And so this is true for fructose and a lot of other ketoses. So that's going to end today's lecture on carbohydrates. So what we've discussed today is what a carbohydrate is, the stereochemistry of carbohydrates, the various stereoisomers of carbohydrates that exist and their naming, and their naming conventions, the difference between D and L monosaccharides, and finally the way in which those ketoses and aldoses can undergo a hemiacetal reaction to produce ringed forms, which are the more stable forms. Next time, we'll begin to talk more about the reactions of carbohydrates and the reactions that they undergo. I look forward to seeing you next time.